Hey guys, welcome to part 2 of chapter 11, Fundamentals of the Nervous System. We left off on graded potentials. We had just really covered graded potentials. We were talking about membrane potentials in general and what they were. And again, it's a separation of charged particles across a gradient. We talked about resting membrane potential, which was about negative 70 millivolts, give or take, depending on the cell. And now we're going to be talking about action potentials. Action potentials are the star of the show. Reason being is that they're capable of transmitting over long distances without decay. But before we get into that too much, let's just run through these bullet points. So an action potential is a brief reversal of membrane potential. So remember, rest is negative 70. The peak of an action potential is actually 30. So that's a 100 millivolt difference from negative 70 to 30. So that's a big move. It's a brief but big reversal of membrane potential. My air just kicked on, sorry. Hopefully that's not too loud. That brief reversal is broken up into 3 phases. We're going to go through each of these in detail, but for now just know that they're called depolarization, repolarization, and hyperpolarization. Why are action potentials so important? They do not decay with distance. If I need to wiggle my big toe, I need an electrical signal that can travel about a meter or so from my brain to my lower spinal cord, synapse with the second neuron, cause a new action potential to be formed that travels from my lower spinal cord to my big toe, to my extensor hallucis longus, whatever muscle I want to activate. So we need a pretty powerful signal, or rather a signal that self-propagates, and we'll explain that as well. They last about 3 milliseconds. Now what that means is if I were to put an electrode in the middle of one of your axons, let's talk about a neuron that's we're going to use to wiggle our big toe. So the neuron goes from our lower spinal cord to a big toe muscle. How do you like that for anatomy? A big toe muscle. Extensor hallucis longus, brevis, flexor pollicis. Flexor hallucis, one of those, right? So I have an electrode in the middle of that axon. So I can watch that action potential drive by the electrode because the electrode is stationary and the action potential is going to travel by it. And if I were to have that electrode plugged into a little oscillator that measured current changes, I would get an image that looks like this. And from start to finish, it'd be about 3 milliseconds. And you can see that on the graph. So it starts right about here. I hope you can see my cursor. It starts right about here before the depolarization spike, and it ends after hyperpolarization. So we're at like from 1.5 to 4 is about 3 milliseconds. You can also call this a nerve impulse. So let's now break down what is actually happening in each of these 3 phases, and then let's try to turn this graph that really doesn't mean anything into a visual image of how an action potential works. So baby steps. It's kind of a little conceptual, a little abstract, but we will get there. So the next 4 slides right here. Each slide has a title, and it's the title of one of the 3 phases of an action potential. We've also included rest here. That's why there's 4. Rest isn't technically a phase of an action potential, but you start at rest, so it's important to know what we're starting from. So we're going to look at all 4 of these. I normally draw this on the overhead. We are going to try to draw it on a PowerPoint. Wish me luck. So here's what I'm going to do. I'm going to draw a patch of cell membrane. Let me be a little bit smarter about this. All right, on each slide, I'm going to have 3 channels. I'm going to have a voltage-gated sodium channel, a voltage-gated potassium channel, and then a potassium leakage channel. So let's draw the potassium leakage channel. I'm just drawing a protein channel. This isn't necessarily anatomically accurate. Then I'm going to have a voltage-gated sodium channel. And I'm going to draw a gate here, closed. And then I'm going to draw this, which we'll explain later. It looks like a little ball and chain. And then let's draw a voltage-gated potassium channel. We can label them, so this would be a potassium leakage channel, a voltage-gated sodium channel. It's hard for me to write, so hopefully you can write on your paper. Voltage-gated sodium channel, and voltage-gated potassium channel, and I need to make a gate. So it's a gated channel. Okay, so we just drew resting state. All gated sodium and potassium channels are closed. That is true. Potassium leakage channels are always open. There's no gate. So if you guys think back to the video from part 1, you remember that I don't know if I would draw this if I were you. It's going to get messy. So you know, this is these are little potassium ions, K pluses. They were leaving, right? Do you remember that? They were leaving down their concentration gradient. But at some point, there was an electrical gradient that started to form. The inside became negative. And it started to pull them back in at the same rate they were being pushed out. So that equilibrium is called resting membrane potential, and it happens at about negative 70 millivolts. So let's add something to our picture. Man, my AC is loud, I'm sorry. Let's draw, going along with the photo, a graph. So what does an electrical, if we had an electrode right here, what would it look like? It would look like this, negative 70. And as time went by, the flat line would just continue because this is an unstimulated neuron. So the inside is 70 millivolts more negative. That's what these are, minus signs, than the outside. So the outside is relatively positive compared to the inside. Okay? All right, that is rest. That's technically nothing new. We've started talking about the channels a little bit more. Now let's go to depolarization. Depolarization is kind of a big deal. Something big happens. What is that? Well, it's a brief reversal of the membrane from negative 70 all the way up to 30. So 100 millivolt change. And that change is brought about by current flow. And that's what we're going to draw. We're going to draw that current flow and what channels that current flow utilizes. My drawing is deteriorating somehow. Okay, do you guys remember what these are? This is a leakage potassium channel. This is a voltage-gated sodium channel. And this is a voltage-gated potassium channel. Now, during depolarization, the sodium channels are open. That's what causes depolarization, is the sodium channels open. Sodium is a positive ion. This looks like a W the way I drew this. Let me fix that. Sodium is a positively charged ion, and we talked a lot about this last quarter. I don't know how much we've said it this quarter, but it's also highly concentrated outside of the cell. So this is outside. We'll call this extracellular up here. It is very hard to write with the mouse. Extracellular and intra. Okay? Cellular. So there's a lot of sodium outside. So sodium then, when this channel opens, it rushes down its concentration gradient. It's also, if you think about it, it's also rushing down its electrical gradient because the inside is negative. So sodium just floods the axon. Remember, this is just one little patch of the axon. We could continue this drawing forever and draw thousands of each of these channels. We're just looking at one little segment. I hope that makes sense. Imagine now we zoom out on our microscope and that this is an axon, you know, and the cell body's up here, and we have some axon terminals. We're just zoomed in like on one little cross section here, one little piece of membrane. That's what we're looking at. Anyway, okay, so sodium floods in, potassium channels are still closed. Some of them actually will probably start to open, but let's keep it simple. It's closed. And then this little guy here that we drew last time is still looks like that. So what did we just draw? We drew sodium entry. Okay, so let's draw our graph again. So rest is done. Okay, so that's rest, which was at -70. Some time has gone by, and then this happens. Okay, 30. And that happens due to sodium entry. So this is phase 2, depolarization, versus phase 1, which was rest. And what drives this depolarization? Sodium entry, due to voltage-gated sodium channels open. So we haven't even talked about why this happens. We're just talking about how it happens. But what we'll learn is that if there's enough graded potentials acting on a neuron cell body, an axon hillock, if there's enough depolarization graded potentials, we can start to move the resting membrane potential from -70. We can start to move it up a bit. And at -55, voltage-gated sodium channels open. That's what I just drew here. And once that happens, we get an action potential. There's no stopping it. Okay? But I'm getting a little ahead of myself. That's depolarization. Let's look at repolarization. So here we go. We got to do it. We got to draw it again. I'm not going to label them. I think you guys know. So that's the potassium leakage channel. You go ahead and label them, though, if you'd like. This is the voltage-gated sodium channel. And this is the voltage-gated potassium channel. Leakage channels are always open. That never changes, so we can just leave that alone. Voltage gating sodium channels, this gate is open, but let's read this carefully. Sodium channels are inactivating. So it doesn't say that these channels are closing. Instead, there's a second gate. We've been drawing it every step so far. That little ball comes here and it inactivates the sodium gate. Oops. So even though it's open, it's really functionally it's closed. These main activation gates are open, but the channel is closed. And that's just because it takes some time to reset. So it has to inactivate first and then these gates close later. Anyway, potassium channels are open during repolarization. So what does that mean? Well, where is potassium highly concentrated? Potassium is highly concentrated inside. Despite the fact that it's always leaking out and being pulled back in at rest, it's in equilibrium, overall there's always much more potassium inside the cell than outside. So when you open a bunch of channels, a bunch of potassium channels, it still rushes out. So potassium rushes out during repolarization. Sodium channels are closed, so nothing's happening there. So what does that look like on our graph? So rest was in, we'll do in pink here. And then depolarization was phase 2. Now we're in phase 3, which is repolarization. During repolarization, the voltage-gated potassium channels open. Potassium rushes out. Potassium is a positive ion, so the inside of the cell becomes more negative. In case I wasn't clear, this graph is representing the inside of the cell membrane. So remember, at rest it's -70, and then it goes all the way up to 30 at the peak of depolarization, and then in repolarization it comes all the way back down to rest. Depolarization was due to sodium entry. Repolarization is due to potassium exit. On the test, sometimes I use the word influx and efflux, and it confuses people. So phase 2 is due to sodium influx, which means entry, and phase 3, repolarization, is due to potassium exit or efflux. Okay, hyperpolarization. We don't need to draw this one, it's pretty straightforward. Basically, let's just draw the graph. So here we go. Rest, depolarization, hyperpolarization. So 1, 2, 3. And then it's debatable whether rest is a part of this. So I'm not going to be like, hey, what's phase 2? I'm going to always ask you a specific question about, hey, what channels are open during hyperpolarization? What is potassium doing during repolarization? What is sodium doing during depolarization? I'm not going to make you memorize numbers. I don't think that's useful. So, and then here we go, hyperpolarization. So hyperpolarization, some potassium channels are still open, and then the sodium channels are now reset or resetting, where the activation gates are closing and the inactivation gates are opening again. So functionally, sodium channels are still closed. Leakage channels are always open, but during hyperpolarization, some of the voltage-gated potassium channels are still open. So we overshoot our target. We go below rest, and then we come back to rest. So here we are again, back at rest. That should be a flat line. That is one action potential. So I don't know if that makes any sense yet. It's okay. We're going to explain it more, and then we'll probably, in a couple more slides, even do a quick video. But before we get there, let's talk about threshold. I mentioned it briefly earlier. Threshold is the voltage that opens voltage-gated sodium channels. You have to hit threshold in order to generate an action potential. It's about a 15 to 20 millivolt membrane depolarization. So if it's 70 millivolts at rest, then threshold is around negative 50 to 55 millivolts. Okay, so what in the world does that mean, you might be thinking? I'm going to endeavor to hopefully make it make sense. So let's do our graph here. It's not a ton of room, but here's our graph. So we're an electrode. Say we stick an electrode into the axon hillock. That's the trigger zone. So if we can bring that area to threshold, we'll shoot an action potential down the axon. Okay, so the axon hillock is key. All of these incoming electrical signals are coming in from the dendrites. Those are called graded potentials. So let's say here we are at rest, negative 70. And then say right here I have another axon coming down and synapsing there, and maybe it synapses here and synapses there, okay? And that particular neuron, say it's coming up from our brain. Let's say it's an excitatory neuron. That means that it's going to open a channel that either lets a positive ion in or a negative ion out. It's going to make the inside more positive, because remember, if we can move the membrane 15 to 20 millivolts in a positive direction at the axon hillock, we're going to make a new action potential. So anyway, this junction is called a synapse, so we'll call this the presynaptic neuron and the postsynaptic neuron. So if the presynaptic neuron sends an action potential that opens chemically gated sodium channels, because these axon terminals release a chemical called a neurotransmitter, and chemically gated sodium channels open up, that means sodium is going to enter these dendrites. Now, one action potential is not going to do anything. But say we send 180 over the course of 1 second, and then let's say there's another one over here doing the same thing, another neuron, and it's sending 100 a second. That's a lot of sodium entry. So these dendrites are being flooded with sodium, and that sodium is going to diffuse down its concentration gradient. It's going to move down its concentration gradient. Just like if you dropped a sugar cube in water, it would dissolve and diffuse. Well, if enough of that sodium diffuses over to this area, the axon hillock, I'm up on my graph again, the axon hillock is going to start to become more positive. Okay? And say negative 55 is right here. And then let's say the stimulus stops and we go back down to rest. That's a subthreshold stimulus. Nothing happens. No action potential is formed. Okay, let's say now we have another neuron. And now they're all, and in reality, there'd be thousands of these. Some excitatory, some inhibitory. And this line would be all over the place, depending on what's going on. But let's say now these are all excitatory. Okay, so they're all excitatory. This one's letting sodium in, this one's letting chloride out. They're all having a positive influence on the postsynaptic neuron. Then we might make it to threshold. And what happens when we hit threshold in the axon hillock? We get depolarization. And I don't have enough room to draw it. To go all the way up to 30, and then it would repolarize, hyperpolarize, and then come back down to rest. Okay, we would shoot an action potential down the axon. That would be a threshold stimulus. So, to put that in practical terms, let's say we're looking at a pain neuron, a neuron in your index finger that senses pain, and you touch a warm coffee mug. That would be a subthreshold stimulus. Your temperature receptors would be firing, telling you, hey, this is warm, but your pain receptors would not be. It'd be subthreshold. But then you say you touch fire, you touch a flame. That would activate your pain receptors. That would be a threshold stimulus. And your neuron would send action potentials to your spinal cord and then to your brain telling you, hey, this hurts. And the response would be to remove your finger. And that would be a series of action potentials that would coordinate that whole process. That tug-of-war occurs over the axon hillock. And if we can get the axon hillock to threshold, an action potential will be produced. And that is this idea of the all or none phenomenon. With a graded potential, you can get a little one, a big one, you can get a big one, then a negative one, then a positive one, depolarization, hyperpolarization, not with an action potential. An action potential is all or none. If you hit threshold, an action potential will be sent down the axon. And so your book uses a great analogy. It's the match analogy. And the analogy is this. Say you take a dry stick, say you're camping, and you hold a match to it. And you hold the match to the end of the stick for 5 seconds and you remove the match and the stick did not catch on fire. Did you change the stick at all? You did. You definitely did. You warmed it up. That would be an example of a graded potential. You warmed it up. You could warm it up a little, you could warm it up a lot. But at some point, there's a temperature that will cause it to light on fire. That ignition temperature represents threshold. So if you hold the match there long enough, it's like a graded potential. It gets warmer, it gets warmer, it gets warmer, then you take it away and nothing happens. That's a subthreshold stimulus. But then you put it back and it gets warmer and warmer and warmer, and then you hit threshold. Say it's 200 degrees, I have no idea. The mat or the stick ignites and catches on fire. Now if you remove the match, the stick is still on fire. Removing the stimulus does nothing, because once you hit threshold, you get the response, even if you remove the stimulus. So that analogy works pretty well because neurons function the same way. You can depolarize the axon hillock a little bit, but unless you get to threshold, nothing happens. But once you hit threshold, once you get to negative 55, an action potential is produced and then propagated down the axon. And that's actually what we're going to talk about next. What does propagation mean? We've been looking at a stagnant graph, right? We've been looking at what an action potential looks like as it passes by an electrode inserted into an axon. Let's zoom out a little bit and look at the whole axon and see how it works. So I think this is a good good model here, but it's just a model. So what I'll probably do after this, I'm going to probably stop this video, try to find a good action potential animation and edit it in here. So with any luck, that will go pretty smooth. We'll see. Anyway, so here is a segment of an axon. Just to give you an idea of where we're at. Okay, just here's the axon hillock, let's imagine, in the cell body, in dendrites, okay? And then the axon continues down, let's say to the foot or something. Okay, so we're just looking at a segment here. So an action potential was triggered here, and we see sodium entry. That's why it says Na+, and you can see the inside of the membrane is more positive. So this segment right here is at the peak of depolarization. It's up here. It's at 30 millivolts. I can't draw off the screen. Oops. So right there, it's up at 30 millivolts. It's at this part of the action potential. This is all at rest. All the other segments are just a flat line at rest, okay? So let's imagine then that what we're doing, this graph is a representation of an electrode that's plugged in right there. So that's what it would look like. And then though, all the sodium is rushing in. Now remember, this area is positive. It's like 30 millivolts. And it's positive because there's so much sodium. Well, sodium is an ion that diffuses, so it's naturally going to diffuse over here, right? It's going to spread out. And as it does so, it makes this area more positive, and it brings it to threshold. So once it comes to threshold here in blue, now voltage-gated sodium channels open up there, and sodium rushes in there. Okay, that's how it spreads. And then the next thing happens, and it spreads some more, and then it spreads some more. But our electrode's right here, so what do we see? Now we see potassium rushing out, which is repolarization. And then here we're back at rest. Okay, so we're back at phase 1. So phase 1, 2, 3. They didn't illustrate hyperpolarization, but 4, and then back at rest. So this is this is a mess, I'm sorry. But I'm just trying to give you an example that when we draw this graphical representation, we're just looking at the electrical events at one patch of membrane. But in reality, an action potential is a fluid thing. It spreads. And it spreads down the cell membrane, down the axon, by triggering the next patch of membrane to open its voltage-gated sodium channels. Now, that's different from electricity. I mean, this is electrical current because it's charged particles in motion, so it is technically electrical current. But when you think about a copper wire carrying electricity, that is conduction. If we think about like a direct current, not to get out of the woods, but a solar panel with a wire coming into your house, there are actual electrons moving and being conducted like water through a garden hose, maybe. Let's use that as an analogy. This is very different. This is much more like dominoes. Okay, so if there's a domino right here, right? What happens is this knocks this domino over, which knocks the next domino over, which knocks the next domino over. It's a chain reaction of membrane depolarization, followed by a chain reaction of membrane repolarization. So you get this wave of sodium influx traveling down the axon, followed by a wave of potassium efflux. So it's a little bit different. That's why they like to call it propagation instead of conduction. Because every new patch of membrane is a brand new action potential. So this action potential triggers one here, triggers one there, triggers one there, triggers one there, and it's like a domino effect. It eventually reaches the end of the axon, and something happens, a neurotransmitter release, for example. Okay, so what's interesting about all that is, what that means is every action potential is identical. They all, within the same cell type, they all look identical. So this action potential looks like the same as the one at the end. So if they're all identical, how do you tell the difference between something that's, say, 50 degrees? So if I put 50-degree cup of water in one hand and a 100-degree cup of water in the other hand, neither one would hurt. 100-degree water is not that warm. It's not Celsius, like boiling water. I'm talking Fahrenheit, like jacuzzi water. So if you had a 50-degree in one hand and a 100-degree in the other hand, how do you tell the difference? If the receptors in your 50-degree hand are firing the same type of action potential as the receptors in your 100-degree hand. So the way it works is that the receptors code for stimulus intensity by changing the frequency of action potential creation. So what that means is the 100-degree hand, the temperature receptors are going to be stimulating the axon hillox of those neurons more powerfully. The graded potentials will be more intense. And that intensity is going to cause action potentials to be triggered more rapidly in those receptors. So your spinal cord is receiving action potentials at a greater rate in your 100-degree hand than the 50-degree hand. Okay, and there's some limits to that. There's this idea of a refractory period. An absolute refractory period is it's like the max. There's a minimum amount of time that has to transpire between action potentials. So there's a time between each action potential that you cannot make another action potential, no matter how strong the stimulus. That's called the absolute refractory period. So there's some limits to that. So your brain then kind of knows the range of potential action potential speeds or frequencies. So let's say you have a temperature receptor transmitting at 100 action potentials per second. Your brain may interpret that as that is very hot. You're touching very hot water. Maybe not painful in the case of a temperature receptor, but very hot. Versus a hand that's simulating 20, you're going to say, okay, that's lukewarm or cold. I don't know the exact, I don't. You'd probably have to talk to someone very versed in neurophysiology to get to that level of detail. But the point is that you use action potential frequency, or rather your brain uses action potential frequency to determine how strong a stimulus is, not the intensity of the action potential, not the amplitude. So there are some test questions like action potentials vary in A, amplitude, or B, frequency, and you would say B, frequency. Or how do you code for stimulus intensity? By varying action potential frequency. Or what would likely have a higher action potential frequency? Stepping on temperature is the easiest. So putting your hand in 50-degree water or 100-degree water. Well, 100-degree water would be a more intense stimulus to a temperature receptor. Okay, we'll do a quiz in class. I'll post one next week so you can kind of get an idea of how I ask questions. So I'll probably just publish it on anponline.com and you guys will take it. So stay tuned for that. And just, I don't know if I already told you this, but because it's online, it's just going to be open note, open book, not even timed. The purpose is for you to get practice for when we take the test, because the test will be timed. We might even do it in person depending on what's going on in the world at that point. But so don't stress about the quizzes, but try to come somewhat prepared so you get something out of it. Okay, what influences propagation of an action potential? Well, what influences conduction velocity of an action potential or propagation velocity? Well, axon diameter does. So the larger the diameter of the axon, the more quickly the action potential will move. Also the degree of myelination. So so far we've been looking at non-myelinated axons when we've been drawing our receptors. So the action potential has to stimulate each new patch of membrane. So each one of these is a brand new action potential. We get new sodium influx at each point of the membrane. That takes time. That's called continuous conduction. Saltatory conduction is a little bit different. So we have the same axon. Let me get rid of this stuff. But now we have, let's say we're in the peripheral nervous system, so we have Schwann cells. Okay, so what's interesting about these Schwann cells is now all we have to do is create an action potential here. The sodium diffuses rapidly to the next section because this is completely non-permeable to ions, and then we get a new action potential. So it looks like the action potential is jumping, and that means there's fewer action potentials, so it actually is much quicker. So that's called saltatory. Saltare means to leap, so it's leaping conduction. It's not really leaping, but it kind of is because it's kind of skipping segments of membrane. So you don't have to do as many individual action potentials. So it's just a quicker style of propagation. So the fastest axon is going to be a thick, heavily myelinated axon. And we see those in motor neurons and sensory neurons, where if you hear a rattlesnake or see a rattlesnake, you want to be able to move quickly or move your foot. Or if you step off a curb and you don't know the curb's there, you want to feel it and react as quickly as possible so you don't hit your head on the ground. That's a kind of an evolutionary or a biological imperative or motivation for us to develop heavily myelinated motor and sensory neurons so that we can react and stay alive. And you'll see that other things not so much. You know, if you need to relay a feeling of an upset stomach to your brain, that can take a few seconds. We don't need to devote resources to myelinating those neurons. So anyway, axon diameter and myelination are the primary methods for influencing conduction velocity. And there are different fiber types, A fibers being the fastest, C being the slowest, B being in the middle. So this is a range of axon diameter and myelination that I will not quiz you on, so don't worry about that. The synapse. So we need to talk about the synapse. So far, we've just been talking about action potentials traveling down this axon, but in reality, that's just a small part of the picture. Typically, an action potential is a lot has occurred before the action potential is even triggered. One neuron has had to bring another neuron to threshold. So we'll call this the presynaptic neuron and this the post. And all of these little guys right here are synapses. So there's two, and actually there's three. Your book talks about two, but there's actually three. Oh boy, can you read that? That third letter's an O. Axo-axonal. So there's three types of synapses. Axodendritic. So here's a synapse between an axon terminal and a dendrite. That'd be axodendritic. Axosomatic. These are axosomatic. And you can even have axo-axonal. You can have a synapse right on the axon hillock. So, and here we have the presynaptic and then the postsynaptic neuron. So this presynaptic neuron is trying to exert influence over this postsynaptic neuron. And in reality, there'd be thousands. It's not going to just be two, right? But this is trying to influence control over the next neuron. And then this cell body of the postsynaptic neuron is taking in all this information from all these synapses. And you have depolarization, you have hyperpolarization, meaning excitatory and inhibitory synapses. And there's this tug-of-war over the resting potential of this neuron. And if you hit negative 55 in the axon hillock, you get a new action potential. So in the case of pain, okay, so say this is a pain receptor, okay? That's not what a pain receptor would look like, right? And you touch a hot stove, burning hot stove. There's going to be an action potential created right here. Better believe it, right? Because these dendrites are stimulated very strongly. But then that's not enough for you to feel pain. This pain receptor neuron then needs to stimulate this neuron here, okay, in order for you to feel the pain, react to the pain, etc. And it's much more complicated than this. There'd be branches going up to your brain. There'd be connections on interneurons and the spinal cord and motor neurons. So it's more complicated, but this is just the basic idea. So how strong these synapses are is going to be based on a few things. Number one, what is the frequency of action potential propagation? So if we're touching a hot stove, it's most likely very high. So maybe this neuron is sending 100 action potentials a second, which is kind of the max. But what else influences the strength of the presynaptic neuron's ability to influence the postsynaptic neuron? Synapse location. So here we go. Axodendritic synapses are the farthest away from the action potential. So let's say they're allowing sodium to enter the dendrite. That sodium has to diffuse quite a way to make it to the axon hillock. Axosomatic, these ones are a little bit closer, right? So these synapses, they don't have to, that sodium doesn't have to diffuse as far. And then the strongest is the axoaxonal, because that synapse is right on the axon hillock, which is the area we care about. If we bring that area to negative 55, we get an action potential. So those will be the strongest. So axodendritic weakest, axosomatic in the middle, and axoaxonal are the strongest. There are two types of synapses. Very few of them are electrical. We're going to be talking about the chemical synapse, which means these neurons aren't hardwired together. There's a chemical reaction that occurs. Not really a reaction. We release a neurotransmitter into the synaptic cleft. So let's talk about that. Actually, let's stop here. Let's do this. we're going to go watch 2 videos. We're going to watch a video on an action potential, and then we're going to watch a video on a chemical synapse. Okay, we're going to watch, hopefully, if I can find them. If it all works out good, if I'm able to edit it together, you're going to see that first video right now. All right, guys, sorry I can't make this full screen. It kicks me out of my recording. So this match is representing some type of stimulus, I suppose. In reality, it'd be ion flow from the adjacent membrane, but whatever, that's fine. So they're showing you a buildup of positive ions. So this is still a graded potential because we haven't reached threshold. So we have positive ions building up, building up, building up. At some point, threshold will be reached, and voltage-gated sodium channels will open. Boom. That's what they're showing you there. So now we get that big depolarization spike. This is an action potential. Some of those ions float to the adjacent patch of membrane, depolarizing it. And eventually that area will be depolarized so much that threshold will be reached there as well. And then we're going to get another big depolarization spike when those voltage-gated sodium channels open up. And this is going in super slow motion. This blue area is a repolarization area. It's also going to be a refractory area, meaning that we cannot form a new action potential there yet. It's still resetting. But as the action potential moves on, so too does the refractory area. Anyway, so this domino effect continues all the way down the axon, and it propagates down the axon. The reason why it can't go backwards is because of this refractory area. So it causes a one-way transmission of action potentials. So I hope that helped a bit to visualize. If not, you can just leave me a comment on Canvas and I'll give it another shot. Okay, let's now from here, let's go to the synapse video. I'm going to try to find a good video showing a chemical synapse, which is the standard synapse between neurons. All right, guys, I suspect that this is going to be pretty good. It looks fancy. So here we have, let me pause it really quick. So this is going to be the end of our presynaptic neuron. It's just one axon terminal. And then this is going to be a dendrite. Okay, I'm assuming that's the end of a dendrite. So we have a gap between these two structures called the synaptic cleft. So what we're going to see is it will probably be a glowing light or something to represent the action potential reaching the end of the axon terminal. So that sodium depolarization domino has reached the end of the axon terminal. And then what's going to happen is that calcium will be absorbed. Okay, so that action potential triggers calcium channels to open. Calcium rushes in, and calcium influx triggers vesicle release. So there's going to be little bags of neurotransmitter in the end of this axon terminal, and they're going to be released. Then that neurotransmitter will diffuse across the cleft and bind to chemically gated channels. So let's say it's acetylcholine, and it binds to an acetylcholine channel. That acetylcholine channel is going to open up and allow, let's say, sodium in. In that case, it'd be excitatory, because sodium would rush in, bringing this dendrite's membrane potential up. Okay? And then if you get enough of that, you'll get an action potential in the axon hillock. Now, there are no voltage-gated channels in the dendrites or the cell body, so no matter what happens here, even if we get this membrane potential up to 50, right, from -70 to 50, no action potential will form because we need voltage-gated sodium channels to form. So the axon hillock is always where the action potential forms, okay? I don't think I mentioned that yet. But anyway, back to the synapse. Let's see what happens. Okay, so the action potential arrives. I'm not sure what's going on here with that glow. Okay, there we go. So action potential arrived and calcium enters the axon terminal. Vesicles filled with neurotransmitter are released. So there's the neurotransmitter, and then it binds, comes across, and binds to chemically gated channels where it allows, say, sodium to enter. causing a graded potential to form. And that's it. All right, I'll see you guys back at the slides. All right, we are back. So we just watched a video about this slide. Just a quick recap. What happens? How do we transfer information across a chemical synapse? Well, we have the presynaptic axon terminal and the postsynaptic dendrite or soma, or we don't know exactly where this synapse is, but the action potential reaches the end of the axon terminal. Voltage-gated calcium channels open, calcium rushes in and causes neurotransmitter release. Neurotransmitter diffuses across the cleft and binds to the chemically gated channels, the postsynaptic receptors. Binding opens those channels, creating a graded potential. And then once very quickly after that action potential from the presynaptic neuron has reached the end of the axon, that neurotransmitter is reabsorbed. Unless there's continuous stimulation, then that neurotransmitter stays in the cleft. And that's part of how you influence stimulation strength, is by frequency of action potentials. But anyway, the neurotransmitter is reabsorbed and the effects are terminated. So we've been talking a lot about excitatory synapse and inhibitory synapse. So it works like this. So let's draw a presynaptic. This is a axon with a little axon terminal. And here is a postsynaptic dendrite, let's say. Let's draw a few dendrites. Okay, let's draw a few more axons. Okay, so these three axons are going to be fighting over this axon hillock's membrane potential. So what is an excitatory synapse? An excitatory synapse is going to cause this patch of membrane to become more positive. So let's say this is an excitatory neuron. And then this is an inhibitory. And this is excitatory. So inhibitory neurons cause hyperpolarization. They cause the membrane potential to become more negative, making an action potential for less likely to form. Excitatory synapses make this area more positive. They cause depolarization, and the more depolarization here you get, that spreads down to the axon hillock. So excitatory versus inhibitory. Excitatory causes depolarization, inhibitory causes hyperpolarization. So there's a lot at play here. There's whether or not the synapse is excitatory or inhibitory, how close the synapse is to the axon hillock, and then what type of summation is occurring. So let's do this. Let's say that we have 3 excitatory action potentials in a row. Okay, E1, E2, E3. You can label those if you want. So now we're going to put an electrode in the axon hillock, okay? And we're at rest. But then these excitatory synapses arrive. So we get a little, so this is -70, and this is -55. So here we go, we have E1, E2 arrives, E3 arrives. Maybe we hit threshold. Then we get a big action potential, right? So this is from E1, the first excitatory action potential, E2, E3. It is so hard to write. So 1, 2, 3, 1, 2, 3. Do you get how that might work? That's called temporal summation. That's temporal summation because it's summation over time. Now let's say we have 1 inhibitory. And then maybe we have 2 more. Oh, I'm sorry, that's excitatory. So we have 1. Is that an I? Yeah, why did I do that? So let's say that's, excitatory 4, and this is inhibitory 1 and 2. So it might look more like this. We have 1, 2 excitatory, and then 1, 2 inhibitory, and then 3, 4 excitatory. Okay, this is summation over time and space. So that's a combination of temporal summation and spatial summation. So spatial summation means summation from multiple sources. Temporal is over time. So the combination of temporal summation, spatial summation, excitatory synapses, inhibitory synapses, and then the location to the axon hillock, that all comes into play over the integration of graded potentials that's going on in the cell body and the axon hillock. So in this case, this drawing is a subthreshold stimulus, right? So it will eventually just return to rest because we didn't hit 55. But if we had hit negative 55, it would spike. We'd get depolarization, repolarization, hyperpolarization, and the action potential would travel down the axon. Don't worry about synaptic potentiation and inhibition. I think we've got enough on our plate. Same with this. We're not going to go through classifying neurotransmitters. We may actually be done. Let me see. Yeah, I think we're good, guys. So I just want you to basically know the differences between a graded and an action potential. I want you to understand what integration is. Integration is this tug of war over the cell body and the axon hillock. Where does integration occur? In the cell body and the axon hillock. I want you to understand excitatory versus inhibitory. A graded depolarization in terms of a graded potential, hyperpolarization in terms of a graded potential, but also understand the events of an action potential. Phase 1, depolarization. 2, repolarization. 3, hyperpolarization. What channels are open during which phase of the action potential? Okay, there's a lot here. There's a lot to unpack. It's kind of, this is the most kind of conceptual and abstract chapter, these part 1 and part 2 so far, that we've covered together. So don't feel bad if you don't quite get it yet. We'll have a practice quiz. We'll probably schedule some time to talk about this on a Zoom call, and we'll have a practice midterm. We're going to do all kinds of practice. We can even talk about it in-person lab, if you guys are feeling confused. Okay, so I'm going to go ahead and edit this and get it posted. Thanks, guys.